Hi, everyone. Thanks for joining us from the Scientists in Every Florida School Initiative. This is a program led by the University of Florida Thompson Earth Systems Institute. I am Brian Abramowitz. I'm one of the K-12 Education and Outreach Coordinators, and I'd like to welcome you to our third monthly Nature of Science webinar. Our mission at the Scientists in Every Florida School Initiative is to build long-term collaborative relationships between teachers and scientists, and bring current research and data into classroom lessons that are connected to Florida Sunshine State standards, and in doing so, connect the scientists with every Florida school. Today, we are joined by Dr. Mariela Pajuelo. She received her PhD from the University of Florida in zoology, and as a Peruvian biologist, she spends much of her time studying the feeding behavior of sea turtles. She's here today to talk to you about some of the research that she's done and its relevance to your classroom. I'm very excited that Dr. Pajuelo is here to discuss her area of expertise and how it may be used in your class.
Okay, well, hello everybody. Let me just give you a little bit of more background about me. Yes, I am a Peruvian biologist, and ever since I started my studies in Peruvian University, I became fascinated with the ocean. And you couldn't help it living next to the Humboldt Current, whose high productivity supports one of the largest fisheries in the world, that of the Peruvian anchoveta, which is this small fish that you see here. But I became more interested in the larger marine animals that live there. And so in order to study them and learn more about them, I work with different kind of people, from government agencies to fishing communities. So I got to know and share knowledge with fishermen, their kids, and even go aboard small-scale fishing vessels like the one from this little fisher kid that you see here in northern Peru. But today I want to talk to you about how science and technology can help understand more about the sea turtle behavior and allow us to obtain information that will help for their conservation. A little bit of background about sea turtles. Well, what you see here is Arcalan. This is a fossil sea turtle and is the largest sea turtle that ever lived. And so sea turtles have been roaming all ocean basins for more than 100 million years. They've been here for so long, and they have even outlived dinosaurs. And they're still here with us today, even though they're face so many threats like fisheries bycatch, this incidental capturing fishing here. So we could say that the history of sea turtles is one of survival and resilience. But let's talk about the most common sea turtles that we see here in Florida beaches, the loggerhead turtles. They're found all over the world, as you can see here in this map of the distribution of the nesting sites. The larger the circle, the greater the number of turtles that are nesting there. And as you can see, the southeastern USA, and in particular the Florida beaches, are host of one of the largest nesting aggregations for loggerheads in the world. And so when I came to Florida to do my graduate studies around 2007, we were experiencing a dramatic decline in the number of nests that these loggerhead turtles were laying in Florida beaches. No? And at the time, people were wondering what's going on, what's happening to them? Why aren't that many turtles coming to nest? But before we answer this question, let me give you a little bit of background about sea turtles. So after loggerhead turtles hatch along the Florida beaches, they're going to embark in one of the longest migrations across the Atlantic Ocean to these developmental areas where they're just going to grow and develop for up to 14 years. Time after which they're going to go back to the other side of the Atlantic and stay in coastal waters. They're going to spend around 20 to 30 years until they reach maturity. And they're going to migrate again, but this time they're going to go to their breeding areas. And the female is going to lay their eggs, and after they're done mating, everybody goes back to their coastal foraging grounds. And they do that every 2-3 years. The cycle repeats. So knowing that the turtles spend so much time out there in these feeding areas in the open ocean, people were thinking, well, maybe there's something going on in their feeding areas that it's impacting them. Some threats like fisheries bycatch, maybe they're getting caught in fishing gear and dying. And so that's why not many turtles are coming to the nest. And so that's one of the questions that researchers wanted to answer, including me. But how do you follow a sea turtle after it laid its eggs and goes back into the water to its foraging area? How do you monitor this large animal and to this vast ocean out there? And so what people use is telemetry. This is one of the most powerful tools that allows to remotely monitor wildlife. And it involves attaching these electronic tags to an animal. So these are small devices that can record and store data, not only from the animal, so the location where the animal is going out there in the water, but also about their environment. They can record and store data, temperature and depth, so we can learn more about the diving behavior of the animals, for instance. Some of these devices need to be recovered in order to access the data. By others, like satellite tags, are gonna transmit information in real time via satellite. And because there are many satellites around the world, we can have information about the movements of animals in the water over 1,000 kilometers. No? So what you see here is a turtle that is ready to be fitted with a satellite tag. And so once it goes back in the water, every time it surfaces, it's gonna transmit data to the satellite, and that information is gonna be useful for us. The satellite tags can last for several months, but there have been instances where they've lasted more than a year. So that's what researchers used to learn about what the loggerheads that were nesting along the US were going. And so one of my collaborators intercepted the sea turtles in the nesting beaches, fitted them with their satellite tags, and then followed their paths. No? The dots that you see here are the locations of all the turtles that were tracked. And as you can see, they're using mainly coastal waters, all the way from New Jersey down to the Bahamas and the Florida Keys. And not long after that, another one of my collaborators did the same with the male turtles. He captured them in this breeding area and then followed its movements. You can see each red line here represent the movement of a turtle, and the numbers is where they stop and stay and just decided to, that was their foraging area. And so you can see that they use the same areas, coastal waters, and even go to the Gulf of Mexico. So this was great. We learned where the foraging areas of the loggerhead turtles were, but there's only so many turtles that you can track with this technology because it's very expensive. Each of these satellite tags can cost up to $5,000. So there's an alternative approach to learn about the migration of animals and identify the foraging areas, and that is using the stable isotope analysis. So this analysis is considered now one of the most useful tools for researchers to study not only the animal movement, but also their diet. And so stable isotopes are these chemical biomarkers that are naturally occurring in the environment. No? And so the isotopic signatures of different food webs are going to vary. And this variation is going to be reflected in the tissues of the organisms via its diet. So if we analyze the tissue, the stable isotope values of the skin, the blood, the muscle, or even the hair of an animal, we can have the isotopic signature of the food web where this animal was. And so this way, we can track the movements of animals if they're moving through different food webs. The most commonly used stable isotopes in the marine environment are carbon and nitrogen. And so when I started my research, I did a thorough literature review. And I found that in the North Atlantic, the stable isotope values of nitrogen here on the right of zooplankton vary. In the north, you can see that they're higher and lower in the south. And the opposite was found for the carbon stable isotopes. They were higher in the southern part, and lower in the northern part. But the resolution that you can see here is coarse. There was not much variation along the foraging areas that loggerheads were using. So something else that I realized was that these areas that were used by the loggerheads fell within 3 distinct biogeographic regions. So biogeographic regions have distinct characteristic environmental conditions and also biological conditions. So for instance, you can see here, this is a map of the sea surface temperature. The waters here up north in the Mid-Atlantic Bight are cooler and highly productive than the waters here in the south, which are warmer year-round and not that very productive. And also the diversity of benthic invertebrates, which is the preferred prey item of loggerheads, is higher here in the south than going up north. So with previous work that I have conducted, I knew that all of these characteristics would affect the isotopic signatures of each of these food webs. So I decided to analyze the stable isotope values of the tissues of these loggerheads that were tracked to these different foraging areas. And this is what we found. And you can see here, we just need a tiny little piece of sample from the turtle, skin sample that we collected from the dorsal area of the neck. This is a green turtle, but you get the idea. And so the turtles that were using different feeding areas had different isotopic signatures, and this was great. I was able to combine satellite tag with stable isotope and validate the use of this technique. And this way I could infer the foraging area of any turtle that I would find. So I would find out where these turtles were actually coming from. And that's what I did. Using the stable isotope analysis, I was able to infer the origin of more than 200 turtles that were nesting in Florida beaches, which was great. I just need to go out there to nesting beach, find the turtle, and get a tiny little bit of sample, skin sample. But sometimes you go to nesting beach and this is what you find. Turtle came and went, and so you miss your chance to get your precious sample. So what do? Well, at the time that I was doing my research, we were lucky that another of our collaborators were doing this neat genetic project in which they wanted to ID genetically through DNA each of the nests that were laid in nesting beaches north of Florida. They were using the eggshells. So we asked for the other parts of the egg yolk. And we found that we do not need to sample the mother, the nesting turtle on the beach. We can just use the egg yolk as a proxy of this isotopic signature of this turtle and figure out where this turtle was coming from. And so one of our recent studies that just been submitted for publication shows this, a compilation of all the samples collected along the beaches of the United States. And you can see that we have collected yolk samples and skin samples. And in some cases, we've analyzed these isotopic values of more than 200 turtles. That's a lot of turtles per nesting beach, no? And we see this clear pattern. Turtles that are nesting in northern nesting beaches are going to use these highly productive and cooler waters in the Mid-Atlantic Bight. And then turtles, as you go south, turtles that are nesting down in Florida beaches tend to use these warmer waters around the Bahamas, the Florida Keys, and Cuba. And not only that, if we sample turtles throughout the years at a same nesting beach, we can see how the proportion of turtles coming from the different feeding areas changes. And in this case, in this nesting beach in Wausau Island in Georgia, we see that the majority of turtles that are coming to nest are coming from this feeding area in the northern waters. So now the next step that we did as well, and I'm not showing it here, is that because you know where these turtles are coming from, you can see how the use of these different feeding areas can have an effect on their body size. Are turtles going to different foraging areas have different sizes? Do they grow bigger or smaller? Do they lay more eggs? Do they lay more crutches during the nesting season? And so the stable isotope data analysis has helped us not only understand the trends in abundance in the nesting turtles at the different nesting beaches, but also has helped us to learn a little bit more about how the different foraging areas can have an effect on their reproductive biology. And so by the time I figured out where the turtles were feeding, the numbers of these turtles starting to go up again. So we were not facing this pessimistic view that they were going to go extinct, but they were going up again. And so, to be honest, the exact reasons of this dramatic fluctuation in the nesting numbers of the loggerheads in Florida is still unknown. But many people think that is due to conservation efforts that began decades ago. So in 1978, loggerhead turtles were listed as threatened species. So they've been protected ever since. And so after that was 40 years ago. And a decade later, in 1987, the shrimp trawlers were required to use this turtle excluder device, which is a metal grid that's placed in the net, so turtles can escape if they get caught when the shrimp trawlers are fishing for shrimp. No? So perhaps what we're looking at right now is, because the turtles take up to 30 years or more to reach maturity, what we're looking at now is all the success of these conservation efforts that took part in the past. And so sea turtles, like many other highly migratory species, are, as you can tell, are difficult to access throughout their lifetimes. We're lucky to be able to work with nesting sea turtles when they come to shore, but what about the other life stages? So as more advances are performed or conducted on these technologies, such as the telemetry that I show you, or stable isotope analysis, or other natural tracers, our ability to improve our understanding of the behavior of sea turtle is going to improve. And so we're not only going to be able to acquire information about what they're eating, where they're eating, but also assessing trends in abundance of these animals. And ultimately, all this information is crucial for managers to make effective management plans for the conservation of these animals. So I think I'm done with my presentation, and I'm ready for Brian to continue with
Okay, well, thank you so much. I really enjoyed learning more about your work, and it's really fascinating to know how conservation scientists do the work, identify problems, and then try to make action plans because of that and what they can do about it. So my colleague Stephanie Killingsworth and I came together after speaking to Mariela and her work, and tried to figure out, well, what does this have to do with the classroom? How can we form connections to what teachers in K-12 classrooms are doing? So we came up with a few learning standards that you can certainly use in your own classrooms. But of course, after hearing that, there are so many more that come to mind, whether it's within biology and the tissues of the animals, or whether it's in chemistry and isotope compositions, and how scientists use that to identify certain factors within the animals or fauna. But one of the biggest one that of course comes to mind is this last one. Identify, research, and collect a data set on a topic, issue, problem, or question using age-appropriate technologies. And the way that Dr. Pajuelo was able to use the technology, and what was it called? Telemetry?
Yeah.
Telemetry. Yeah, to use that and understand the conditions that turtles were going through once they entered an environment that scientists had a much more challenging ability to enter and observe the animals, but all that data back to them in a really meaningful way. Okay, next slide. Okay, so we're gonna jump into our question and answer. When I announced to some teachers that we were going to have Dr. Pajuelo here talking about sea turtles and the work that she's done with them, some teachers have reached out saying that they do have questions, or their students have questions about sea turtles. And this is going to be our opportunity to hear those answers. So our first question during this time is going to be, can you explain how you take the nitrogen and carbon samples from the turtle's epidermis or skin?
Yes, that's a great question. Actually, I didn't go into much detail because I didn't want to extend my talk, but it's not that very hard. Well, you need to get the sample first, and all you need is a tiny little bit of sample. Even you saw the sample, the skin sample that I took from the turtle. That's a lot. I could run I could do 2 runs of nitrogen and carbon stable isotope analysis with that sample. So what you need to do is just clean it very carefully, and then you chop it in little pieces. You dry the sample in an oven for a day or so, and then you put it in these little tin capsules. And when it goes in the tin little capsules, then you take it to this mass spectrometer, which is in here at UF. We have one in the geology department, and then it's gonna combust the samples, and it will give you results about the isotopic composition of carbon and nitrogen that you can later use. So it doesn't take long. The run itself could be done in a day. It's just so difficult sometimes if you're working with sea turtles to find the samples, and I was glad to have so many collaborators at different nesting beaches that would be willing to let me use the skin samples they collected.
Thank you. We received another question from a teacher. They said, with nest temperature of some reptiles determining sex, are there concerns for sea turtles due to climate change?
Yes, definitely. Sea turtles the sex is determined by temperature, and so the higher the temperature, the more females that are gonna be hatching. And so you could see that in the future, as the temperatures go up and the nesting beaches get hotter, we're gonna have more and more females. And we still don't know. I mean, if we still have a few males or even just one male, we could think that maybe that would be enough to actually preserve the population, but we are not sure yet what the ratio is, the best ratio that we need to have males and females. But it's certainly not a good thing to only have females, because eventually the population is going to die out if you don't have those males to make them more babies.
Sure, of course. What is the biggest kind of sea turtle?
So the biggest sea turtle is the leatherback turtle, and it's an amazing turtle. Not only because it's big, it can be as tall as 1 meter 80 centimeters. That would be like 6 feet, a little bit higher than 6 feet, and it's not like the typical sea turtle with a hard carapace, but it has a softer carapace, and it's called that's what it's called, leatherback, and it's kind of black, and it's amazing to see it out there in the nesting beach. I personally haven't been able to see a nesting turtle. I've seen the hatchlings, but I'm willing to take the chance to go down here in Florida, because they nest here in Florida, and just experience that awesome thing to see them nesting.
And our last question, what's the biggest threat to sea turtles today?
So the biggest threat of sea turtles today, I know you've heard a lot about plastics and pollution, and you've even seen that impactful video about the strawberry pulling out of the nostril of a sea turtle. Even though we know that that is one threat that sea turtles face, I would say that the biggest threat that they face nowadays is fisheries bycatch, which is when turtles are incidentally captured in the fishing gear. So for instance, turtles can get caught in shrimp trawlers. Likely in US waters, you are required to use these turtle screw devices, but in other countries, it's not required. Many turtles die in shrimp trawlers. And they also get hooked in long lines, long lines with hooks, and sometimes they just drown also in this drift net or fishing nets that are just floating in the ocean. So I would say that that's the main threat, but luckily people are doing a lot about trying to mitigate the impact of the fisheries. There's different technologies that are being used right now to prevent to reduce the mortality of sea turtles and their capture in these fishing gears.
Thank you. And from all of us here at the Thompson Earth System Institute and all of us at the Scientists in Every Florida School Initiative, we'd like to extend a huge thank you and our gratitude to Dr. Pajuelo and all of you teachers watching. Thank you so much for tuning in. Please join us again next month on December 9th at 7 p.m. We will hold a live webinar where paleontologist Victor Perez will talk about his work with megalodons. Thank you.